Welcome to your first online lecture for Biology of Plagues in Bio 104. This is a two-part lecture. This is part A. I'm also posting part B. I will also post the accompanying PowerPoint slides. They're already posted as both a PDF and a PowerPoint file so that you can print them out and take notes into them and follow along as you listen and take notes on this lecture. Please take notes very carefully. I will periodically check your notes to be sure that you have viewed the online lectures, and the content of these lectures is quizzable and useful for completing your homework assignments, just as in-class lectures are. If you have any questions, please email them to me or come by and ask me in person or ask me in class. The goals for this part of the lecture are to talk about what a cell is and what the bare minimum is that a cell must possess, some detail, and also to learn some detail on what is in prokaryotic and eukaryotic cells, talk about what parts are present and what those parts do, and we're going to talk about who first observed cells and how. Please be sure to take careful notes as if you were in class as you go through this lecture file. A cell can be defined as the most basic unit of which all organisms are comprised or made of. They are relatively tiny. I'll talk more about that in a moment. A microbe, by definition, is an organism that exists as a single cell. In contrast, multicellular life, such as the plants and animals, consist of organisms where one organism contains billions of cells. Cells can look very different from one another, but they all have common features. Examples of differing cell appearances are shown here in the variety of cells depicted in the figure. Bacterial cells are relatively simple. Bacterial cells are shown here. You saw cells like this, although perhaps the images weren't quite as clear. This one's been photo enhanced in your recent lab activity. You'll note that bacterial cells, although they're quite simple, are still metabolically complex and still able to achieve all of the functions required of cells. And as you'll see in a few moments, you'll learn that they have basically the same parts as those seen in eukaryotic cells. All of the other cells shown here are examples of eukaryotic cells. This is an example of a plant cell, or a group of plant cells, and the other three are examples of cells that are found within our own body. Skin cells, neural cells, or a neuron, and an egg cell, human egg cell. So what I like looking at when I look at or think about cells is I actually really enjoy looking at cells from the human body, such as the three shown here, and I really find it fascinating that even though these are all from the same organism, their appearance varies so drastically. You want to remember that each of them is specialized, which allows it to perform a particular body function. So that neural cell has very long axial processes which can carry electrochemical signals, and these reach across the entire body through the nervous system, allowing us to control our bodies, respond to the environment, sense pain, feel pleasure, and so on. Our skin cells are there to protect us from the outer environment, to keep moisture in, to keep pathogens out, and they are relatively flat cells and slough off continually. So there's a high rate of turnover for the skin cell and a low rate of turnover for the very highly special neural cell. And the egg cell has its own particular design so that it can participate in reproduction. They're all very different looking, but they all have common features, as do all cells. That's what we're going to talk about now. Most cells are microscopic, meaning that they are smaller than the 200 micrometer. A micrometer symbol looks like this. It's a funky little U and then an M, and that's a micrometer. It's a metric unit of length, very, very tiny. There are a million micrometers in a meter. A meter is about the distance from one of your shoulders to the tip of one of your fingers. Anyway, most cells are microscopic, meaning they are smaller than the 200 micrometer width that the human eye can see, unaided by any sort of magnifying device. This image, the image shown in this figure, or in this slide, gives us a feeling of scale, taking us from the atomic level, over here on the left, to the tissue level, over here on the right. So cells fall in here. Okay? So here's a red blood cell. It's 6 microns which equals 6 micrometers. A micron's just sort of like shorthand for micrometer, wide. So you can't see that because it's smaller than 200 micrometers, the typical size something has to be for us to see it with the unaided eye. So the only way we can see those structures is if we magnify them, and that's what we use microscopes for. So similarly with bacteria, typical bacteria might be, anywhere from 5 to, 100 micrometers in length, going from the very small to the very large, and we can't see them with unaided eyes. This is true for most cells. I should add that the link takes you to a site where you can play around a little bit, and it's a really interesting interactive tutorial that will give you a bit of a sense of the scale of some of these really tiny objects and relate them to objects that you're maybe more familiar with. So let's consider now what the bare minimum is for cells. Like how simply can we talk about them and still be having a meaningful conversation? What do all cells have, and basically why do they have it? So all cells have cell membranes, which are often referred to as the cytoplasmic membrane. This bounds the inside and outside of a cell. In other words, it dictates what is inside and what is outside. And importantly, it controls what goes in and what goes out. So it's protective of the contents inside the cytoplasm. The cytoplasm is a watery fluid that fills the inside of a cell. Also in the cytoplasm are salts, proteins, sugars, and other molecules. Kind of like a soup. All cells also have chromosomes, and the chromosomes are made out of what is called DNA, or deoxyribonucleic acid, DNA. Okay? This is really important molecule, and it's important because it contains what we refer to as the genetic information, or the blueprints, that allow proteins to be made. And once you make proteins, proteins are a group of molecules that accomplish all of the work of a cell. That work is metabolism or energy processing, locomotion or moving around, transport, getting things in and out of the cell, and then a variety of other functions. That should be an R in there and a space. I apologize for that typo. So without proteins, you can't do cell work. So you can't reproduce, and you can't metabolize. So you're dead, basically, from a genetic perspective and a cellular perspective. So you need proteins. So the information to make the proteins is maintained in DNA. So all cells have to have both of these things. All cells also have common ways of dealing with energy, chemical energy, within the context of the cell. And the common so-called energy currency of cell is a chemical called adenosine triphosphate, or ATP. Okay? All cells have ATP. These are common to all living things and to all cells. That's the bare minimum. So, a long time ago, scientists came up with a list of qualities or characteristics that basically define life, or can be used to answer the question, is this or is that alive? Is it a living thing? Is it an organism? And these qualities are shown here. They're cellular organization, energy production / processing, I would say. Okay. Reproduction, irritability, or the ability to respond to the environment, and the ability to undergo growth and development, even at the level of a cell. Okay, so even a microbe can undergo growth and development, some sort of specialization here and there. Now in red I've put cellular organization because it's what pertains to this particular discussion that we're having. So all life consists of cells. Therefore, all life, if you look at its most basic building block, you see units that are organized in characteristic patterns, those of cells. They all have membranes encasing water-based solutions and cell structures. They all have DNA in the form of chromosomes containing genetic information. And they all have proteins and also structures called ribosomes, which I didn't mention a moment ago, that are used to convert genetic information into proteins. So in other words, it is the structure and the function of a cell, it is cellular organization, that actually allows for the qualities 2 through 5 to occur. Without cellular organization, 2 through 5 could not exist. I have discussed the idea of 2 fundamental cell types with you, but I want to focus really in this lecture more on how these cells are fundamentally similar instead of being different. We're going to take a tour through the composition of a prokaryotic cell next, and then we're just going to spend a moment or two looking at eukaryotic cells. But what I want you to hold in mind is I want you to be looking for the common elements. What do all cells do? What do all cells have? And then that will actually help you better understand these 2 different patterns, because you can ask, well, how do prokaryotes achieve the function known as maintaining genetic information? And how is that done in eukaryotes? How are they the same and how are they different? So I want you to be thinking about making a note that addresses that in your study notes. Let's look at the basics of a prokaryotic cell. So this could be any bacterial cell or any archaeal cell. Because our class focuses on infectious disease, let's talk about bacteria. So here's a simple bacterium shown here. We've looked at this cartoon a number of times already this semester. If we start from the outside in, let's have a category and let's call it appendages. These would be attachments to the cell's surface. I don't need that there. So the types of attachments to the cell's surface that bacteria have are flagella, pili, and fimbriae. The flagellum is shown here. Okay, it's that long kind of brown structure. The colors are sort of meaningless in this context. They're just for making the cartoon pretty and easier to distinguish one part from another. Bacterial flagellum are very important in motility or movement. Other appendages that bacteria have are pili and fimbriae. And shown here on this particular figure, these little green structures are pili. Pili is plural. Pilus is the singular. Okay. There are no fimbriae shown here, but fimbriae would be even smaller appendages. Bacteria use pili and fimbriae for a variety of reasons. They use them in some cases for certain forms of motility or movement, and they also use them for defense and offense. For example, pili can be used to secrete toxins. That would be an example of an offense function. Okay? A defense function might be the use of pili, actually more likely fimbriae, or pili for attachment to environmental surfaces. For example, attachment to the epithelial cells lining the human body. That would be a defense function from the point of view of the microbe, because it would allow the microbe to stay in place in an environment that provides it with food. All of these extracellular appendages are made out of protein, different types of protein, and some of them are known as pathogenicity factors. Okay, so a pathogenicity factor is an aspect of a microbe that allows it to be pathogenic, or in other words, able to cause infectious disease. And how could these things be pathogenicity factors? Well, let's think about secreting toxins. If you've got some aspect of your cell structure and you're a microbe, and it can be used to secrete a poison that harms your host, then that will make the host sick. And when the host becomes sick, usually there's some benefit to the microbe. So maybe some host cells degrade and the microbe gets more food. But in any case, it's something about the microbe that makes the host sick. So the microbe is using that function to be pathogenic. Okay, let's consider some other areas of prokaryotic cells. Moving in, let's think about the cell envelope. So not really shown on this, well actually it is shown in dark blue down here. We've got the word capsule. So many microbes, many bacteria in particular, make capsules. They make them out of carbohydrates or certain types of sugars. Carbohydrate and sugar you can think of kind of synonymously. And they make capsules in order to protect themselves. They provide protection from drying or desiccation, also attack from phagocytes. So phagocytes would be animal cells that can engulf and destroy an invading microbe. Okay, so if you've got a capsule, you might be able to protect yourself from that fate, and also from viruses. Viruses act as cellular parasites, and although there are many viruses that attack humans, there are also viruses that attack plants, and there are viruses that attack bacteria. A capsule can help to protect a bacterium against such attack. Capsules are also helpful in allowing bacterial cells to attach both to one another and to surfaces. So because of these features, capsules can be a pathogenicity factor. Remember, I defined a pathogenicity factor in the previous slide. So if you think back to that protective function, phagocytes, which I defined a moment ago, are really important parts of our immune system. If you've got a bacterium and it's invading you and it's got a capsule, and our immune phagocytes cannot engulf and destroy that bacterium, then they can't get rid of it from your body. And so that makes the microbe more able to multiply and infect you. And so that makes capsules a pathogenicity factor. Also part of what we think of as the cell wall, or of the cell envelope, is the cell wall, which I discussed in a previous lecture. I mentioned in that previous lecture that the cell walls of bacteria are made of peptidoglycan, a fusion macromolecule consisting of amino acids and carbohydrates, and that the role of the cell wall is primarily to protect cell from water stress. Protect them from blowing up, basically, or lysing from water stress. Also part of the cell envelope is the plasma membrane. The plasma membrane is made out of lipid. It's a special kind of lipid or fat called phospholipid, and it serves to control what goes in and out of the cell. It bounds the cell, and also in bacteria, it is the location of ATP synthesis, the energy currency for the cell.